Dr. Eric Danner from the School of Medicine, who's going to talk to us about his audiovisual aids that he's developed to improve students' learning in systems-based embryology.
Right, so thanks, Katrina. Thanks, Katrina and Jade and colleagues for inviting me to speak today. So I'm going to talk about this developing audiovisual aids to improve student learning on systems-based embryology. So this is some innovations we're using in the Department of Physiology and the School of Medicine here in Trinity. But I go right back to how this started. It started really 10 years ago, and how I became interested in teaching innovations. I am a neuroscientist, so my research is based on neuroscience. So this is somewhat of a side interest. So how it started is really 10 years ago. In my first lectureship, I was an anatomist in University College Cork, and I worked there with several colleagues, John McKenna, Andre Tully, John Crine, and it ran cadaver-based teaching. So we ran cadaver labs for teaching medical and dental students, where we had cadaver stations and osteology stations and clinical stations. So working with the students, we heard their feedback every day about what they were doing and what they were looking for. And we learned at this time, and from dental students in particular, they really were learning certain things off by heart and then forgetting them the next 2 or 3 days afterwards. And one of the things they were learning off by heart is a typical topic of cranial nerves. And these are 12 pairs of nerves in the head and neck region. And they're involved in many different physiological functions in the body. So in terms of vision, smell, balance, and hearing. And students are using various different mnemonics or acronyms to learn these off by heart. So we thought, how can we address this? And when I looked at literature, we found that there was some evidence that different 3D models or MIME, for example, were being used to educate students in the area of cranial nerves. So we thought at that time, what else can we do in that cohort of students who are teaching to enhance their learning experience? So we asked the question, does a cranial nerve animation enhance student learning? And this was my first PhD student moved here alone. So we developed a PhD around this in teaching innovations, particularly around anatomy for dental students. And we published a number of papers working with John Crine and Andre Toulouse on different tooth morphology apps and different embalming techniques to enhance student education. But I'm going to touch very briefly on this one. So this was relates into the project we're doing now in Trinity somewhat. So it's an animation tool which we published a few years ago that's supplemental dental students learning. And what it was is it's a 4 to 5 minute simple whiteboard animation. And this individual, I have about 30 seconds of it, see if it plays in a second, fingers crossed, that basically this guy is going to see an apple, pick it up, bite it, swallow it, and it works through your 12 pairs of cranial nerves in that process. So it's trying to encourage students when they have their lunchtime that day to think about using the nerves in an everyday scenario. So we try to make it a little bit cute as well.
My name is Jimmy. Today I'll be talking to you about the 12 pairs of cranial nerves in the head and neck region of the human body. These nerves are part of the peripheral nervous system, and all nerves originate mainly from the brain and exit the cranial cavity by passing through foramina and fissures. We use these nerves subconsciously in nearly every action or movement that we do. Some nerves have sensory function, such as the olfactory, optic, and vestibulocochlear nerves. Other nerves have motor function, such as the oculomotor, trochlear, abducent, accessory, and hypoglossal nerves. And some nerves are in fact mixed, having both sensory and motor capacity, like the trigeminal, facial, glossopharyngeal, and vagus nerves. While these nerves have several actions, in the following animation, we'll focus only on the function of the nerves related to this scenario. I'm hungry. Apples. The second cranial nerve, the optic nerve, carries the sense of vision from the retina to the brain.
So you see the idea of it. So we worked through the 12 nerves in that scenario. And then we introduced that over 3 years into the student cohort of dental students, and we found that it improved their learning of that specific topic compared to students that didn't have access to the animation tool. And that was through the Blackboard platform in Cork at that time. So that was published in this article. Right. So then onwards to the current study. So I I joined Trinity in 2015, and a part of my role, I've been acting director for 9 years of the BSc in Human Health and Disease, which is a 4-year degree program here in Trinity in the School of Medicine. It's a direct entry program. It's about 35 students in each year of the class, and there's the 4 years of the program. I'll just draw your attention to this specific module here, developmental embryology in year 1, a junior fresh year. So this is a 5-credit module out of 60 credits that the students undertake. And we put it in the first year of the Human Health and Disease program because it adds a basis of understanding for large human form of function modules that these students undertake in second year with medical students. So it is quite heavy content for first year, but we put it in first year because it adds a foundation of knowledge in the health sciences. And I started to focus on educational tools in this module because I had selfishly had full control over it, as I teach all the content within it. And you see some of the content on early embryology and assistance-based embryology in the human body. And anybody that works or studies in this area, you know that it's extremely complex topic, but it's a fascinating topic. So students were telling me when I started teaching this module that, they really enjoyed it mostly, but they were struggling at times with certain concepts. So what I did then in 2015 or 2016, I looked at the literature, and there was some evidence of different educational tools in embryology that were being used to enhance student education, and multimedia tutorials and different cardiovascular tools. So I asked the question, can an embryology animation similar to what we did for cranial nerves be developed for the human health and disease students in Trinity? What we did, I was lucky to receive a Dean's Award for Teaching Innovation in 2016. And that allowed us then to, in the summer project, then to take Karen Jordan on for the summer project, who was between our 3rd and 4th year of human health and disease at that time. So she had taken the module herself a few years earlier. So working with Karen, we developed then the content of an embryology animation tool for the first year human health and disease students that summer. And we simply did a script and various different actions. We worked with Capsule here at the time in Trinity to develop this and incorporate it onto Blackboard. So we took textbook images, and then I collaborated with a computer scientist, Tamara Bag, in University College Cork still, who then created original images of the textbook contents that we could then use in our admit that person.
Julia.
So we had our own images then that we can use in our innovations. So this is what we did. I will show you 30 seconds of this as well. We developed 2 about 4-minute teaching tools. They're not specifically animations, I'm using the wrong term, but one was for early embryology and one was for gastrulation and one was for neurulation. Now, in context, when I was looking at this morning, and this was 2018, 2019, and since COVID, we're now recording all our content. So it does look, to be honest, slightly behind, but you can see what we did at the time. So this is directly from incorporated into Blackboard.
This animation will summarize the main events in the process of neurulation. Following gastrulation, neurulation takes place in the embryo. Neurulation.
So you can see I had my script down the side that the students could read also, so they had a visual and an auditory component.
This is the process whereby the neural plate firstly develops and the neural tube is created on this neural plate. At day 19, a thickened neural plate in the ectoderm appears, just cranial to the primitive node. On the diagram, the top of the image is the cranial region of the embryo, and the bottom of the image is the caudal region of the embryo. Broadening of the cranial end during week 3 leads to the specification of cells in the ectoderm changing them to neural cells. That is, cells related to neurons or the nervous system. This image represents a transverse section through the neural plate.
Okay, so you can see the idea. Stop that. So it's horrible listening to yourself. Right. So what we did, we designed the study. We did the study over 3 years, where with first year human health and disease students. Where they undertook a questionnaire at the beginning of the study on their embryology knowledge. And then I randomized them to a non-animation group on the left and an animation group on the right. The non-animation group just had lecture content, just listening to me. And on the right, the animation group had these animation these tools on Blackboard for 3 weeks, and then you completed both groups post questionnaires. So I won't go into the data. I'll show you this demographic information, one piece of information from the publication from this. See on the left there, and I should mention Cole O'Connor is a medical student who helped collate this data with me as well. So he's shown an improvement in the percentage correct answers in quizzes in the animation group. And here in the Likert scale on the right-hand side, the animation group ranked animation more highly as an educational tool compared to particularly to textbooks. So that was published last year here in the Annals of Anatomy. And I just mentioned this is a collaborative project with Dennis Barry in anatomy and also Jean Fletcher in biochemistry immunology here in Trinity. Right, so onwards to what we're doing now. I was very fortunate to receive another Dean's Award last year. So we took a similar approach somewhat. So this is William. So William he just finished human health and disease last week, got his final year degree grades. But last summer, he could come back and work with me throughout the summer to develop a new educational content. And we focus on systems-based embryology in this case, based on students' feedback in particular. And Melody and Almadena, who's here as well, are PhD students in my lab, and they've also helped with developing this content. So working with William then we just went through the content. We decided to focus on the development of the respiratory system in the human body, embryologically. And he basically, in a similar process, to develop the content as a storyboard and images. And we worked again with Tamara in University College Cork to develop original images that we can use in our content. And this is it. So we've been using Articulate. Please don't ask me about how to use Articulate. The students know how to use Articulate. I don't. So this is a pilot so far, right? So I'm going to show you where it's at. And we're going to, I've then pilot tested it with the students, and I'll show you their feedback. And then we're going to incorporate that content into enhancing it over the summer and launch for the next academic year. So what you'll see me in 30 seconds here playing around with it. So I'm going to start it first. So let me see if I can pause this. So what you can see is the embryo on the left here, and then you zoom in on part of the respiratory system. This is called the respiratory diverticulum. That's the beginning of your lung forming embryologically. Importantly, we include more interactive components. We have an adult human body, so you can see this in the anterior front view of the embryo. We're looking at the front view of the human. So we can spin it here in this case, like a lateral views and posterior views by using this little toolbar. And you can also use this toolbar to go through certain weeks post-fertilization.
The respiratory diverticulum, lung bone, begins to grow from the ventral wall of the forward. As development continues, tracking isophageal ridges from the bone. So
now you're looking at the right-hand side and starting to spin it. And the left, and you see that the bottom here, the human and adults spinning. So I'm pulling it around here as well. Now flicking on the bottom right through different stages of post-fertilization and time, flicking back and forth. And then the other thing we're trying to do, student feedback said we want quizzes and MCQ content. So here's where we are so far, just filling in the blanks. Okay, so over the last few months, I did a little pilot study. We received ethical approval to do a pilot study on student feedback on this so far. So this is with the human health and disease students. You see that over 4 years who have completed, we have 27 so far. I've just looked at it. Then mostly we incorporate this on Blackboard. They mostly looked at it on their laptop, alone, at home and on campus and throughout the day. There's a lot of information on this, but this is generally positive. So this is a Likert scale. So the higher this is towards 10, it's a strongly agree. It's quite positive in terms of it's of high quality and we enjoyed using the platform. We have negative questions in there in terms of it not being a useful tool. And we have a high score of 9 there, so it helps with understanding course material. And their perception of learning experience, would you like to see more online educational tools? 100% yes, as one would expect. And see, one of them had a difficulty here. So this is, oh yeah, I'll just show you this as well. So other supporting comments, and very easy to use. It's handy to be able to rotate the viewpoints of the diagrams, etc. And this is my last slide. It's a lot here, but basically this is what we're going to use. This is where they're pointing out problems. So you see the percentage correct answer of MCQs, and they would like the scored quizzes. The AI voice is really annoying. So these are things then, this is really helpful in helping us. William's coming back to work in the summer on this to take the student feedback into account. So what we're going to do, we're going to add a original voiceover, hopefully not me again, but add some interactive quizzes and scoring on embryological knowledge. And then for 24-25 academic year, we're going to roll it out across different degree programs, human health and disease into junior fresh medicine, and hopefully in the biological and biomedical sciences program in Trinity as well. And just to acknowledge people, Melody, Almadena, Karen, and William, and Mary O'Neill has been extremely helpful in School of Medicine, and Cole O'Connor, and Tamara Bag, and particularly the support from the Dean's Award for Innovation Teaching.